Time to finish up Chapter 12 with Concept 12.3. This entire cell cycle, of course, has to be well managed and well controlled. Otherwise, things can spin out of control very quickly. And we've talked about cancer a couple of times. Cancer is a good example of when this cell cycle kind of goes wrong. And we'll mention that again toward the very end of this concept. Concept 12.3 says the eukaryotic cell cycle is regulated by molecular control systems. And you'll remember we kind of talked about those phases of interphase, the G1, the S, the G2, and we talked about calling them gap, and we'll talk about why they're called gap and how there are kind of like safeguards in this whole regulation of this entire cell cycle process. The frequency of how often cells divide kind of varies depending on the type of cell. For example, your skin cells, of course, you probably are familiar with the fact that you lose thousands of skin cells every day. So obviously the skin cells have to divide pretty frequently to kind of replenish that supply. Other cells, like liver cells, generally speaking, don't divide in their lifetime, but they can. They retain that ability. And so if there was, say, some particular type of damage or a repair that needed to be made, those liver cells can divide as needed, but they don't continuously divide in the way that skin cells do. Then there are other cells like fully formed mature muscle cells or fully formed nerve cells that for the most part don't divide at all. They rarely divide. So there's not a ongoing mitotic division of muscle cells or ongoing division of nerve cells like there are with some of the others. So the type of cell very much controls or is part of the factor in how often they divide. These differences result from the regulation at molecular levels. And again, we mentioned that cancer cells tend to sort of go out of control, and they kind of go off the rails and escape the usual controls of the cell cycle. And we'll kind of talk about that briefly at the very end. For a long time, scientists weren't really sure of what controlled this process. How did the cell know which step followed the next step and how long to spend on each step? So at one time it was thought that basically the cell wouldn't go on to the next step until the previous step was finished. Like, for example, if you're talking about G1, well, S didn't start until G1 was finished, and somehow the cell knew at which point it was in the particular cycle. So then there were some other experiments, and the experiment that's mentioned in your book on page 238 kind of leads in another direction. The cell cycle appears to be driven by specific chemicals that are present in the cytoplasm. And the way this was determined was they took cells that were at different phases of the cell cycle. So they had mammalian cells. So for example, it could be, I don't know if they specify, but let's just say it's dog skin cells. And they took these mammalian cells, and some of them were at prophase, some of them were at metaphase, some of them were at anaphase, different parts of the cycle. And they fused two cells together. And the cells that they fused together were at two different spots in the cycle. And then they tried to figure out what exactly would occur when they took these two cells that were at the two different phases and put them together. So if you look on page 238, or I'll show you on the next slide exactly what occurred. And then we'll come back to this question that's at the bottom. So let's take a look. So in these different experiments, so for example in experiment 1, they took a cell that was in the S phase and a cell in the G1 phase. And you can see slight differences there. There's in the S phase, remember, is where they have synthesized or replicated the DNA. They fused those cells together. And then a short time later, they came back to take a look. And at this point, both of these cells were now in the S phase. In experiment 2, they took a cell that was undergoing mitotic division, so it was undergoing mitosis. It was in the M stage, and they combined it, fused it with a cell that was in the G1 stage, and when they came back a short time later and looked at them, both of them were in the mitotic phase. Even this G1 cell that haven't even, remember in G1 you haven't even replicated the DNA yet, but it didn't matter. The cell went ahead and started going through mitosis even though it didn't have another copy of the DNA. The chromosomes hadn't been duplicated. So what this seems to indicate is that there are chemical signals within the cytoplasm that kind of kick this process off. And the S signal kind of overrides the G1, and the M signal overrode the G1 here. And so that caused the change that you see. So let's go back to that other question. So that evidence would tend to point towards some form of chemical signal present in the cytoplasm. Let's ask ourselves this. If you look at that slide, how do you think the results of that experiment would be different if each phase was triggered by the completion of the previous phase only? In other words, if there wasn't some chemical signal floating in the cytoplasm, but there was some other method whereby the cell didn't start the next step until the last step was finished, well then how do you think that experiment would pan out? So again, look in your textbook or at this slide here and just kind of do yourself a quick little thought experiment and ask yourself, how do you think these results would turn out if the cell followed a particular pattern where one phase was triggered by the completion of the previous phase? And just kind of think about what you might see in that particular aspect. So going back to this idea of a control system for this whole cell cycle, you have a series of events that have to occur, and they have to occur in a certain way. You can't have the S subphase occurring before the G1, and you need to have everything occur in a certain order, and they all need to be kind of timed out together. So this sequential events are kind of directed by something called the cell cycle control system, and you can kind of think of this as a automatic clock, or think about like your automatic washing machine, or perhaps like your automatic irrigation timer that waters your lawn. Going back to the washing machine has 4 or 5 steps that it has to take to get your clothes clean. And so once you set it and start it, it will proceed on its own, but it has sort of checkpoints along the way. So for example, you set it, the first thing that's going to happen is the washing machine is going to fill with water. And then maybe step 2 is to release the detergent. And step 3 is to wash. And then step 4 is to drain that water. Step 5 is to refill with clean water. Step 6 is to rinse. Step 7 is to drain. Step 8 is to spin. And then we're done. So you got all these steps, and they have to occur in the right order. So what happens if you're running your washing machine and you have disconnected the water? So you have turned off the water supply. Well, in that case, the washing machine may stop at that particular point, and it may not go further because it's kind of got a trigger mechanism or a checkpoint where it knows if something is wrong at this point, we don't continue on. So if you have turned off the water to your washing machine, it probably won't go through all 7 or 8 steps. It will probably stop right early on at the point where it needs water. Well, the cell control system works very similar to that. They have specific checkpoints along the way, and if there are certain conditions that are not satisfied, well, the cell will stop right at that point. And as a matter of fact, the way it usually works is the cell stops at the checkpoint until certain conditions are met and until it gets the signal to go ahead and proceed. So going back to our interphase and interphase and mitotic phase that we were talking about here. So we have G1, S, and G2 of interphase, and then we have mitosis that occurs right in between. So look at these checkpoints that we have. So in the G1 phase, there's a certain point at which the cell basically stops and won't move forward until a certain signal is met or a certain threshold is reached. Once that checkpoint is surpassed, then the cell goes ahead and continues on through S and G2. There's another checkpoint right before mitosis, and then a checkpoint within mitosis to go back to interphase. So you have these particular areas that kind of control the cell, and if something is not right at any of these checkpoints, the cell will basically stop at that point. For a lot of cells, that first checkpoint within G1, that G1 checkpoint seems to be perhaps the most important one. If the cell receives the proper signals at that checkpoint, then it usually proceeds all the way through S and replicates the DNA and all the way through G2 and then goes ahead and goes into the end phase and divides. If it doesn't receive the proper signal at that G1 checkpoint, it actually will exit the cycle and it will go back into a kind of a waiting period, a non-dividing state that we actually call G0 or GO, G0 phase. The G0 phase is actually the phase that most cells are in. That's the state at which they are going about and doing their business. So liver cells are doing what liver cells do, and muscle cells are doing what muscle cells do. but they're not really in the process of preparing to divide. So most cells are actually in that GO or G0 phase, and some of them stay there pretty much their entire lives, like the nerve cells or muscle cells that we were talking about. So in that G1 checkpoint, if the cell doesn't get the right signal that it needs, it will exit the cycle. Otherwise, if it does get the signal that it wants, it will go ahead and continue through S and G2 and then into the M phase where it will divide. So kind of consider that G1 checkpoint, or actually any of the checkpoints, sort of like a light or a traffic signal. If the light is green and the cell receives the information that it needs, that checkpoint is passed and we go on about our business. But if that light is red and the cell doesn't get the proper signal, it exits the cycle and it goes to that G0 phase that we were talking about. There are two types of regulatory proteins. Of course, they're proteins because it seems like pretty much almost everything within the cell, particularly when it comes to control mechanisms, are proteins. There's two particular types of regulatory proteins that are kind of involved in this cell cycle and whether or not the cell proceeds. And these are called cyclins and cyclin-dependent kinases. And of course, they have an initial CDK, cyclin-dependent kinases. Cyclins are called cyclins because they tend to go through a cyclical concentration. These are a type of protein that at certain times within the cell the amount of them is high, and that at other times the amount is low, and they fluctuate with the cell cycle. So they're called cyclins because the population density of these proteins goes up and down and up and down. The cyclin-dependent kinases are, well, we already know that kinases are proteins that phosphorylate other enzymes or other proteins. They take a phosphate group from ATP and they add it to another enzyme or protein to either activate it or deactivate it. And there's a particular one in here called MPF. I think the actual name, or what the initials stand for on MPF, is maturation promoting factor. But another way to think of this, I think a more simplified or perhaps a clearer way, is to think of it as an M-phase promoting factor, because this is a particular cyclin-dependent kinase that triggers a cell's passage past G2 into the M phase. So there at G2, right before we get to the M phase, there is a checkpoint. And whether or not we go past that checkpoint is dependent upon this MPF. This MPF, this maturation promoting factor, or we could call it an M-phase promoting factor, is a cyclin-dependent kinase, and it is controlled by the amount of a particular cyclin within the cell. And if there's a certain amount, it allows this to proceed, and it triggers it past G2 and into the M phase where it goes ahead and divides. So here we have a couple of illustrations to kind of prove our point or tell us what we're looking at. This MPF is actually always within the cell. So this cyclin-dependent kinase called MPF is always there, but it doesn't signal the cell to proceed unless the concentration of cyclin is high. So these two are tied together. And notice this cyclin concentration rises and falls and rises and falls. That's why it's called cyclin, because it rises and falls. And when the concentration rises, it causes this particular kinase to do its job and trigger the cell to go ahead and go into the M phase and divide into two daughter cells. Down below we have another example of this, kind of just a different way of looking at it. So the cyclin is this little purple portion here. It is a particular type of protein, and the MPF is here. So cyclin begins to build up as we move through the cell cycle. That cyclin combines with the MPF, and that allows the cell to move past that G2 checkpoint. When there is enough of this activated MPF, it allows it to say, okay, we have what we need, and that triggers it on into the M phase. Once the cell divides, the cyclin starts to be broken down and degraded, and so now the amount of cyclin decreases, and the MPF is no longer activated, and so then we're into the other parts of the cycle, and then that process begins again. The cyclin builds up, the MPF does its job, the cyclin is degraded, and we continue on around the cycle. So just to clarify, we have this little protein here, which is called the CDK. That's the cyclin-dependent kinase. When it combines with the particular cyclin, we basically have two enzymes or two proteins combining. We call that combined portion the MPF, the maturation promoting factor, or we were calling it the M-phase promoting factor. And then when the cyclin breaks down, you're back to the CDK by itself. The CDK is always there, but only when it combines with the cyclin in the right amount does it trigger the cell to go into the M phase. Let's finish off Chapter 12 by talking about some of the signals that cause all of this to go on. I mean, this is a very complicated cycle that has to occur, and we have to kind of try to figure out, well, how does the cell know when to divide or how often to divide, and what are some of the signals that cause the cycle to start or stop or to begin and end and that type of thing. So, of course, we have a couple of different signals. We have some signals that come from inside the cell itself. Those would be internal signals. And then we have some signals that come from outside of the cell, perhaps from the surrounding cells or from the surrounding tissue, or even from another part of the body through some particular hormone or something like that that has traveled through the circulatory system. So let's look at the internal signals first. There's a couple of different variety of these, but we'll just look at one as an example. One of the internal signals is, for example, the kinetochores that are attached to the center portion of the sister chromatids, the waist that I keep talking about, the portion where it's thinner. So the kinetochores, if they're not all attached to the spindle microtubules, they will actually put out a molecular signal to delay anaphase. Remember, anaphase is where the sister chromatids are pulled apart and then drawn toward the poles of the cell. So if the kinetochores are not all attached to the spindle microtubules, in other words, the sister chromatids are not all attached to the microtubules, they will send out a signal that tells the cell, oh, wait up just a minute, don't proceed until we're all completely ready. So anaphase doesn't continue or doesn't pull the chromatids apart until all the kinetochores are attached to the sister chromatids and they are attached to the microtubules. Because if they did go ahead and pull apart, well, you may have one or two of the sister chromatid pairs that are not firmly attached to the microtubules, and then you would end up with, say, one of the new daughter cells completely missing that chromosome, or the other new daughter cell has two copies of that chromosome now because they weren't all lined up and properly connected. So that's a good example of an internal signal. Anaphase gets suspended or delayed if that signal gets sent out. If the kinetochores basically say, wait a minute, we're not quite ready because not everything is attached and lined up in that center along that mitotic plate. That's a good example of an internal signal. Some of the external signals, there's a couple of different varieties of these. Good example of external signals are some of these growth factors, which are various types of proteins released by certain cells that stimulate other cells around them to divide. So one good example of this has to do with if you get injured or cut or you fall down and scrape your knee. There's a particular growth factor called a platelet-derived growth factor that will stimulate the division of a particular type of cell called fibroblast. And fibroblast is a type of connective tissue that kind of holds skin and cells kind of all connected together. So when you get an injury, like you get a cut or a scrape, you'll have within that injury a part of the blood called platelets. And those platelets will put out this growth factor that will then stimulate fibroblast cells to begin to reproduce and divide and make more of themselves. And that will help form the scab, and that will help form at the injury site connective tissue that will eventually begin to heal that particular site. So here's a good example of an experiment to kind of show that, where they take human connective tissue and cut it up into tiny little pieces. They use an enzyme to digest all of the extracellular matrix, and so what you're left with is just those fibroblast cells that are just kind of floating in suspension. They put those fibroblast cells into two petri dishes with media down at the bottom. If you don't add the particular growth factor, the platelet-derived growth factor, you won't see those cells begin to divide, or you certainly won't see them divide at a very fast rate. If you add the platelet-derived growth factor that is a signal to these fibroblast cells to begin division, and you'll see they soon cover the entire petri dish. Another great example of an external signal that has to do with growth factors is something called density-dependent inhibition. And basically what that just means is when the cells are crowded, they stop reproducing, they stop dividing. So the cells want to reach a particular density, and they don't want to go beyond that. So for example, if there are cells in a petri dish, they will reproduce and divide until they basically fill that petri dish in a single layer all the way across the bottom. They'll form a single layer covering the entire inner surface, and then they'll stop dividing. And if you were to go in and remove some of those cells from the petri dish, that open spot, the cells around that open spot, will start to divide again until they fill in that gap. The way this occurs is that there's a particular protein, a cell surface protein, that basically there's a binding site of this cell surface protein between neighboring cells. And so when you have enough neighbors around you, you have these binding sites that are filled, and that's a signal to the cell that, hey, we're pretty crowded, so don't go reproducing, don't go making any more of us because there's already plenty of us. Another example of some of these external signals has to do with whether or not the cell is attached to something. A lot of animal cells exhibit something called anchorage dependence. They have to be sort of attached to some sort of substrate before they'll divide. If you have some animal cells just floating freely in a vial of culture media liquid, they don't really go through a lot of division. If they are in a petri dish where they have a solid media that they can sort of bind to or anchor themselves to, then they will divide. That's different from when you see cancer cells, and we'll talk about cancer just a little bit because cancer cells kind of break the rules. They don't really exhibit either one of these. They don't really exhibit density-dependent inhibition, and they don't really exhibit anchorage dependence either. And that's a bad thing because cancer cells will tend to grow out of control. Even when there's already too many of them, they kind of ignore the signals, or they don't receive the signals properly, and they will begin to grow out of control. Here's a good illustration to talk about some of those external signals. First, if you have some cells, they really won't start to divide until they are able to anchor to some sort of substrate. So that's anchorage dependence. Then, once they do begin to divide, they will exhibit density-dependent inhibition. So they will fill the whole area. They'll probably fill the whole petri dish, but then they will slow or stop the reproduction and division process. If you were to remove some of those cells in a normal cell, normal mammalian cell, they will fill in that blank. But then, once again, they'll stop dividing. And notice if you look at these cells, they are closely packed together, but they're not in a big lump on top of each other, multiple cells, and that type of thing. Compare that to some cancer cells. Cancerous cells will reproduce out of control, and you'll see that they will go well beyond this density-dependent inhibition, and they will continue to reproduce beyond what's necessary or beyond what's good. Let's talk a little bit about cancer and what causes cells to become cancerous and what that means when cells become cancerous. Basically what we're talking about is a loss of the cell cycle controls. All of those checkpoints that we were talking about and all these signals that kind of control when and how cells divide, those get thrown out the window when we're talking about cancer cells. Normal cells, like a normal mammalian cell, if you have it in a petri dish in culture, it will divide anywhere from 20 to 50 times depending on the type of cell, and that's about it. That's about its lifespan. So it'll divide about 20 to 50 times before it stops, and then it begins to age and then basically die of old age, basically natural causes. Cancer cells don't really do that. Cancer cells don't respond normally to the body's various control mechanisms. So cancer cells, they don't have to get this growth factor as an outside signal. They either will make their own growth factor, or they will get the signal to divide without the growth factor, or they may have a problem in their cell cycle control system, so where they just overrun those checkpoints, and even if the proper checks and balances are or are not in place, they continue to divide out of control. So basically, cancer cells can divide indefinitely if they have a continual supply of nutrients. Basically, you can say that they become immortal. They divide without stopping, and they almost become like living forever, like immortal. There's actually a fantastic book, if you get the chance, if you're at all interested in science and medical science. There's a great book that I read probably about a year or two ago, but it's very well known, about a particular woman. Her name was Henrietta Lacks, and she had cancer. And way back in the 50s, she went to see doctors, and this was at a time she was African American. And of course, back in the 50s in America, things weren't like they were today, and so she wasn't treated very well. Her family wasn't treated very well. Some of her cells were actually gathered and cultured and used by scientists, and are still in use today by scientists. And her family was never notified of this, and they were never compensated for this. So it's a very fascinating story about her and her life and her family, and these immortal cells of her that are still used in research. These cells are called HeLa cells, from her name Henrietta Lacks. And so there are cells that are still reproducing since 1951. So this woman has died. She died many, many years ago, but there are still cells from her body that are around and are being used in medical studies all over the world. And if you were to put all the HeLa cells that have been created, that have reproduced together, it would be literally hundreds and hundreds of pounds of these cells. So it's a fascinating story. If you get the chance, highly recommend reading the story. But basically, these cancer cells become immortal. They reproduce nonstop as long as they have the nutrients to do so. When a normal cell, when something goes wrong, either with the DNA replication process, or there's a variety of ways this can happen, could be an external problem like a radiation could cause this to happen and could damage DNA. When normal cells convert to a cancerous cell, we call that transformation. And when this cell transformation happens, normally speaking, it gets caught by the body's immune system and taken care of. Most cancer cells are caught and destroyed by the immune system, but some basically escape detection of the immune system, or reproduce so rapidly that the body can't get them under control. You have the formation of tumors, which are basically masses of abnormal cells kind of within normal tissue. So if you have a tumor, it's going to be kind of a big mass of cancerous cells enclosed within healthy tissue as well. Cancer cells and cancer tumors can be of two different kinds. One of them is called benign, and that means that the lump of abnormal cells stays in place. It stays at the original site. If it's a benign tumor, oftentimes, while that is obviously scary and dangerous, oftentimes that can be resolved. It can be treated with surgery. The entire tumor can be removed in surgery, or you can use very targeted radiation, which damages the cancer cells worse, damages the DNA in the cancer cells worse than the healthy cells around it, because the healthy cells seem to be able to repair themselves better than the cancer cells. So you still have some side effects, because it's almost impossible to target just the cancer cells with that targeted radiation, but we're getting better and better at actually targeting just the cancer cells. But still, you're going to damage some healthy cells as well. So that's a benign tumor if the cells stay in place and they don't travel throughout the body. Quite a bit more scary is something called a malignant tumor. A malignant tumor tends to spread throughout the body, invade surrounding tissue, and sometimes can even do something called metastasize. When cancer cells metastasize, they travel through the body, and basically there are cancerous cells that get into, say, the lymphatic system or the circulatory system. They move to other parts of the body, and they begin to form tumors in other parts of the body. So malignant tumors require quite a bit of aggressive treatment. Sometimes they require something called chemotherapy, which is kind of a broad-based radiation therapy. And that is the cause of a lot of the side effects of cancer treatment you see, because basically you are irradiating large portions of the body, because these cancer cells have traveled throughout the entire body in many cases, so you can't really target the therapy. Well, what that does is it has an effect on healthy cells too, and it usually has an effect on fast-growing cells the most. So fast-growing and dividing cells such as your hair follicles, which is why cancer patients often lose their hair during treatment. Intestinal cells in your digestive tract oftentimes get affected negatively. So that's why oftentimes people undergoing chemotherapy tend to be very sick to their stomach and they're queasy a lot and they can't eat very well. And then immune cells, unfortunately. So oftentimes chemotherapy really does damage to your immune system, which then makes you susceptible to colds and other types of infections. So malignant tumors can be quite damaging and quite scary because that cancer can spread, metastasize to other parts of the body. Here's a microscope picture of a cancerous cell, and you can see that oftentimes the cancerous cells take on a different shape and dimension. They look different from the normal cells. They tend to grow oddly. Oftentimes the cells no longer work. They no longer do their appropriate job. So let's say, for example, it's a lung cell and you have lung cancer. Well, those lung cells are abnormally shaped and they don't do their job properly, and they're basically just dividing at a rapid pace and sometimes basically taking over portions of the body. So it can be quite frightening. A good example, of course, is breast cancer. I think this month was breast cancer awareness, and there was a lot of pink ribbon activity and that type of thing going on for breast cancer awareness. Breast cancer is a more common type of cancer than some of the others, so that's why oftentimes it gets a lot of awareness. So within the breast you have glandular tissue, and sometimes you'll have a cancerous tumor formed within that tissue. If it's a benign tumor, it stays in place and it doesn't begin to infect the immediate surroundings or travel throughout the body. Sometimes the cancer cells begin to invade neighboring tissue, and to some degree, if it becomes malignant, then it can metastasize, and those cancer cells move through the body, through the lymph vessels or the blood vessels, and they may travel to other portions of the body and begin to establish themselves and grow tumors then in other parts of the body. And that would be the most dangerous thing that could happen, because it's very much more difficult to treat a cancer that has metastasized than it is to treat a benign tumor like this. There is some good news in cancer research and in studying this type of thing, is because we've made a lot of recent advances to where we can more carefully target the radiation therapy so that we're doing as little damage to healthy cells as possible. There's also been a lot of recent advances in understanding cell cycle and what can go wrong, which kind of helps us understand what might cause cancer and how to combat it. What are some of these cell signals that have kind of led to being able to control cancer a little bit better. Also, we've done things like sequencing the DNA of cancer cells. Generally, cancer cells have DNA that is damaged or mutated in some way. So if we can take a sample of the cancer cells from the body and then sequence that DNA, we can perhaps target treatment that is specific to only those cells with that particular type of damaged DNA. And that means we can perhaps have a better chance of eradicating the cancerous cells and doing less damage to the healthy cells and having fewer side effects, and that would be a good thing. So obviously this is a very complex activity. The complexity of this mitotic function of dividing and growing is quite a lot to study. I mean, it's interesting that there is so much that we know, but there's still so much science to do. So just as a wrap-up, this entire chapter was about mitosis. Here is plant cells, a number of plant cells, and you can see these plant cells are in different stages of dividing, going through mitosis. Most cells spend a lot of their time in interphase, which oftentimes can be considered GO or G0, which is the non-getting ready to divide phase. Then they go into G1, and then they go into S, which is where they create, replicate a duplicate copy of the DNA. Then they go into G2, which is the last phase before they go into M phase and divide. In mitosis, we have a couple of different phases there: prophase, prometaphase, metaphase, anaphase, and telophase, and then cytokinesis. Make sure you take a good look at this chapter. Your textbook has some awesome pictures that are very helpful. Go look at the crash course videos or the Bozeman Science videos that I've added, as well as the BioFlix. All of those I think are very helpful. I like this chapter. I feel like it's a little bit easier than some of the other chapters, a little bit easier to understand. I hope you feel the same way. If you have any questions, please, please, please email me or hop online in one of our discussion forums. We will have our regular discussion forum Sunday night as usual, and our usual quiz this weekend. Have a great Halloween. Be safe. Don't do anything too crazy. And for goodness sake, brush your teeth after you eat all that candy. Have a good day. See you guys.